Hello everyone. Today I will be teaching you the Western blotting. What is Western blotting and how Western blotting is performed. The Western blotting, also known as the protein ImmunoBlot. So here's the definition. Western blotting is basically a qualitative and semi-quantitative analytical technique that is used to detect a protein in any kind of the tissue. But remember that you have to separate out the protein. For example, if you want to detect the proteins in a cell, you have to rupture the cell wall and then you will be analyzing the protein using the Western blotting. So, what is this? What is the Western and what is blotting? There are two other techniques that is also known as Southern blotting, that is performed for the DNA, and Northern blotting is performed for the RNA, and hence the name Western blotting, that is for the protein detection. Remember that it is a qualitative and semi-quantitative. Why we are saying that this is a semi-quantitative technique? Because it does not calculate the quantity of the proteins in a given sample exactly. It just gives us some hint that it is basically gives us a hint that the relative quantity of the proteins is greater in the size. Here you can see that the Southern blotting was developed by the Edwin Southern, and also blotting was developed by the James Alvin and George Stark, and Western blotting by the George Stark as well. So these were the types of blotting. Now I will be telling you the steps in the Western blotting. So basically there are uh, almost three steps. You have to separate out the protein first, then you have to blot it on the membrane, and the third one is to analyze it. So here we have the tissue preparation. So how will you tissue preparation means you have to extract the proteins from the sample. For example, if you are going to study the protein from some kind of the virus, you have to extract the proteins from the virus. So next, when we are done with this extracting the proteins, we will be performing gel electrophoresis. And why gel electrophoresis is performed to separate out the proteins. There is another video on the gel electrophoresis. Click on the given link below to watch another tutorial on gel electrophoresis. So for the time being, I will be telling you the next step. The third step is the blotting. What is blotting? Blotting is a transfer from the gel to the membrane. And what is this gel? This gel is the same gel that we have used for the electrophoresis. And finally, there is another sub step after the blotting that's called blocking. I will be telling you what is exactly the blocking. We use a blocking solution, for example, 5% skim milk. And now the finally we will be detecting there are two ways, like you can detect with the two steps or just single step, just like the direct ELISA and indirect. There are also another videos on ELISA, direct ELISA and indirect ELISA. Click the link in the screen to see this. Okay, and finally when we are done, the proteins are on the when the proteins are separated on the gel and we have successfully transferred those proteins on the membrane, we will analyze it. And there are several methods to analyze a protein. For example, colorimetric detection, chemiluminescent detection, radioactive detection, and fluorescent detection. All these techniques gives us some kind of detectable thing that we can detect that there is the protein we are analyzing is present on the sample or is existing on the membrane. So here we go. So how to prepare the sample? Just as I told you, sample can be taken from anything like whole tissue, cell culture, bacteria, or virus. So what do you need to understand and what do you need to memorize? That you will just rupture the cell wall of the cell and then you centrifuge it and you just extract the proteins. You just separate the protein from the debris of the cell by centrifugation. So when you are done with the centrifugation, you have a protein pellet. You just solubilize it into the detergent or salt or buffer to just make a solution of the proteins, so that the proteins can be directly in the further in the upcoming event. So furthermore, we use the antiproteases enzyme, phosphatase enzyme, that is to avoid the degradation and denaturation of the proteins from the cell itself. So just imagine that you are lysing the cell, and the enzymes that are proteases that can hydrolyze the protein are present in the cell. So what will you do? You will just add some antiproteases so that your protein will not be lysed by the proteases present in the cell. And to avoid protein denaturing, we also perform this tissue preparation on a low temperature, so that you know that when the temperature is high, the proteins can be degraded or able to maintain their solubilized structure. So after the first step, we have a solution of proteins. The further what we will do, we will separate out those proteins on a gel, and the technique to separate out the gels is called as electrophoresis. The protein of the sample are separated using gel electrophoresis. See, so you can the gel electrophoresis separate out the proteins on the basis of the molecular weight. And what do you mean by the molecular weight? It means that the conformation and the charge. Greater molecular weight will have the greater charge, and the smaller molecule will have the smaller charge. So ultimately, protein will be separated out by on the basis of their size. And the most common gel electrophoresis for the protein analysis is SDS-PAGE electrophoresis. What SDS means, sodium dodecyl sulfate. It's a detergent that has some effect on the proteins. We will see in the next slide. And SDS, sodium dodecyl sulfate, and PAGE stands for polyacrylamide gel electrophoresis. So we will separate out the proteins. It's that you need to remember that the concentration of the acrylamide determines the resolution of the gel. What does that mean? It means that the smaller proteins, for the smaller proteins, we will be using higher concentration of the acrylamide gel. For example, if we are making the 5% solution for the larger protein, we will make 10% solution for the smaller proteins. So concentration and the molecular weight of the proteins we are going to study is inversely proportional. So sample, when we are done with the gel, we have made the gel successfully, and we put the protein sample into the wells of the gel. And just I'll show you a picture that'll explain you. For example, so we also add a marker. Marker or ladder is a solution of the protein of the known molecular weight. Like we have the bands, and we will compare our unknown protein with the ladder, with the bands of the ladder that will be appearing on the final membrane. So what is the function of the SDS? This is the protein, this is the polypeptide, that is this is basically the secondary structure of the protein. And when we treat the protein, globular protein, with the SDS, SDS will do two things. SDS will do two things. Firstly, it will linearize the protein, and secondly, it will impart a negative charge to the protein. So now all the proteins are linearized and also have negative charge. Remember that SDS page separates the protein on the basis of their molecular weight. So because all the proteins are linearized, no protein is proteins are no more globular or having secondary structures, so they have become the primary structure, so they can be separated out easily. So here is a complete setup of gel electrophoresis. We This is the green one. This you can see the whole gel. This the green one and the light blue color. So these are the wells that is made while preparing the gel. We load the sample here, and then because we know that there are two kinds of stacking gel and running gel. You don't need to understand that, but then these protein will move towards the electrode, positive electrode, because as we have seen that the SDS-PAGE has imparted a negative charge on the protein, so protein will move from the negative cathode towards the positive electrode, and they will be separated out with the on the basis of their sizes. In the bigger proteins will be on the negative electrode side, and the smaller proteins and will be on the top. So this is the glass plates, this is the gel, and this is the spacer. You don't need to understand all this, you just have some kind of illusion. So the third step is the blotting. When we have successfully separated out the gel on the protein, we will perform blotting. What is blotting? Blotting is basically a transfer of the protein from the gel to the membrane. We will transfer the separated proteins from the gel to the membrane. And how will you do? You will just put the membrane on the gel. You have seen the apparatus. You will just separate out the gel from this apparatus, and then you will yeah, here it is. You will, this is a polyacrylamide gel, then the filter paper, and here is the membrane. So we'll put the gel that has the proteins over it, and this is the membrane. So what will happen? Then the proteins will move from the negative cathodes to the anode, cathode towards the anode. Do not forget that you have to place the membrane on the anode side. If you put over here, the protein will not be imparted on the membrane. Remember that while you are making this setup, this is basically the two filter paper to support the gel and membrane. So step was the blotting. The membrane is placed on the top of the gel, and the membrane that is used for this purpose can be made of nitrocellulose, or it could be polyvinyl difluoride (PVDF). So both have their own benefits and advantages, and it depends whether you want to perform your experiment on nitrocellulose membrane or polyvinylene whatever PVDF. So there are two ways that you can transfer the proteins from the gel to the membrane: with the help of the capillary reactions, yet that you use no current, electric, and with the help of the electroblotting. And as I have shown you there, what is electroblotting with the help of the electric charge. So what membrane should you use? What membrane you want to use depends upon the requirement you are performing the experiment. You know why the proteins will transfer from the gel to the membrane? Because of the two things. Because the hydrophobic interactions and the charge interactions between the proteins and the membrane. So we have nitrocellulose membrane that is more fragile and cheaper, and it can not be used repeatedly. And furthermore, we don't need to perform any pre-treatment before using the nitrocellulose membrane, but the PVDF is sturdier, it's thick, and it has to be treated with 95% ethanol before placing it onto the gel. And it is also the costly. It depends upon what kind of the experiment you want to perform. Like you want to perform the only once, then you will use the nitrocellulose membrane, and if you want to perform your experiment repeatedly on the same membrane, then you will use the PVDF. So I have shown this setup you already. This is basically the electroblotting, and in the capillary reactions you will not apply the electric current. Protein will move with the help of capillary reactions. So still this is a membrane, and we separate out we have soaked all the apparatus into a buffer. So when we separate out the membrane, we have nothing on the membrane. We cannot see the proteins have been transferred onto the membrane or not. We cannot see because we have not added any detection. We will detect in the end. So what does the blocking solution do? Blocking solution blocks the non-specific bonding between the antibody and the protein. So what will you do? You will just soak out your blocking solution into the membrane. There are some famous blocking solution like BSA, and skimmed milk that can be used as a blocking solution. And you also mix a little bit detergent of like 2020. So what will the blocking solution do? The dilute solution of the blocking solution attaches to the membrane in all places where the target protein have not attached, and as a result this will reduce the noise in the final product of the Western blotting, leading to the clear result and eliminate the false positives. So I have told you that the detection can be one step or two step. You in the single step you add only one protein, that you only one antibody that is conjugated, and it will bind to the protein or antigen, and on the other side it will be bound to the enzyme or any detectable thing. In the two step you just add the primary antibody that attaches to the antigen, and conjugated secondary antibodies that attaches to the primary antibody. So here you can see that this is the membrane. We have transferred the protein. This is our target protein, and we have transferred successfully the protein from the sample to the. We are here. We have not added the primary and secondary antibody. So what will you do? When you have a membrane, after the blocking solution, you will add the primary antibody. This is called the primary antibody, and primary antibody has bound to the antigen and the secondary antibody, and secondary antibodies body is conjugated. This is HRP. It's called radish peroxidase enzyme that will convert the substrate into the detectable thing. It could be light or it could be some colored product. So this is the two-step analysis. Like if you have the only secondary antibody, this primary antibody will be absent. So this will come over here, and this will be known as only one single step. But single step is not preferred because you need only one antibody that is conjugated with the enzyme and also specific for the target protein. How will you perform the analysis? When you have successfully transferred the protein onto this, and you have added the primary antibody, and you have added the secondary antibody conjugated with some enzyme or some detectable thing. This molecule, this HRP, is an enzyme. This could be anything. This could be a radioactive thing that will discharge radiations and can be detected with the photographic film. So this is the two-step analysis. So remember this picture is very important to summarize the whole experiment in one picture. This was the membrane, this is the target protein, this is the primary antibody, this is secondary antibody, and this is the substrate. This is the enzyme, and you will add the substrate over here, and it could be a chemiluminescent. There are three types of the detection. Colorimetric detection, in which you add a secondary antibody conjugated with an enzyme, and it will solubilize. It will convert the soluble product into insoluble, and it will give some kind of the color. In the chemiluminescent detection, you will add such a chemical that will be broken down by the enzyme attached to the secondary antibody. Will be converted into some kind of other product, plus it will illuminate some light. The fluorescent detection, you add a probe instead of enzyme. You add just a probe that could be excited with the light, and it will just give the radiations. So here is the colorimetric detection. This is the blocking solution. This is the membrane over here, and this is the blocking solution. This is the antigen. This is the primary antibody. This is the secondary antibody, and this is enzyme. Enzyme is converted the substrate into the color product. So this was the colorimetric detection, and this is the chemiluminescence detection, that you have added such an enzyme that you add a substrate that will be converted into the product, and as a byproduct it will give light. Or either you can use a light probe. You just don't need to add a substrate. You just put the primary antibody and just detect the light with the help of the CCD cameras, and you can also add some radioactive element over here that can be detected by the photographic film, and it will give it will give a black circle on the here. So hope you have a great idea about the ELISA test. Please subscribe to my channel. Keep supporting.